So I am honored to. Hello, am I audible? I am honored to introduce Professor Amit Awasthi. Hello. I am honored to introduce Professor Amit Awasthi, an eminent scientist of the nation. To us, he doesn't require any introduction, of course. He is an eminent scientist and currently working as senior professor at Immunology Laboratory, Translational Health Science Technology Institute, Faridabad, India. Now, he has done several great works which are published in almost all the journals of the repute. Published over 100 research papers, including several seminar papers published in Nature, Nature Immunology, dreams of everybody, Nature Communications, Sciences Advanced, and Lancet Infectious Diseases, with a very high H index and one of the highest kind of citations, 14,000 citations. Of course, he has received numerous prestigious awards, including the Innovative Young Biotechnologist Award and the G.N. Ramchandran National Bioscience Award from the Department of Biotechnology, the CDRI Award for Excellence in Drug Discovery Research from CSIR Central Drug Research Institute, the G.P. Talwar Mid-Career Award from the Indian Immunology Society, and the Young Scientist Platinum Jubilee Award from the National Academy of Sciences, India. I am sure we will shortly go through a very exciting journey. Professor Awasthi, please.
Thank you very much for kind introduction, and I take this opportunity to thanks Professor Ganguly for this kind invitation, and Professor Dr. Neerja for hosting me in Chandigarh. So I will be briefly talking about some of the work what my lab is doing, and I think Dr. Rajdeep Dalal who's also here. So the work is done by Dr. Dalal, which is not published, under consideration. Hopefully, it's gonna be out soon. So basically, my lab works on various aspects of T cell differentiation and T cell functions. As you know, T cells, T helper cells come in different flavors: TH1, TH2, TH17. One of the cells which we are focusing on in the lab is primarily produce this cytokine called interleukin 9, and this interleukin 9 is actually one of the cytokine which was identified initially as a T cell growth factor just right after the discovery of IL-2, which is another T cell growth factor. And subsequently, the role of IL-9 is being associated with multiple diseases, including autoimmune diseases, autoimmunity, cancer, allergic inflammation, and extracellular pathogens. So what is the cellular source of this is primarily the T cells which produce interleukin 9, and this interleukin 9 ultimately goes and work on different cell types to cause inflammation in various conditions. So ever since we discovered this subset of T cells in 2008, a lot of people got interested to identify the role of Th9 cells in different disease conditions. As you can see, some of these mentioned in your on the screen: the tumor immunity, Helminth immunity, inflammatory bowel disease. The role of these cells are coming out very big in a inflammatory bowel disease, which I will be talking about in subsequent slides. So my lab is working on various things. Since these cells are so critical, therefore, my lab is invested into identify the kind of transcriptional landscape and the transcription factor or druggable target to suppress these cells in the condition so that we can, cure or provide a kind of drugs for diseases. Since I'm going to talk about the IBD, and IBD, as you know, is kind of a interaction in three different things: micronutrients, that means what we eat, and that actually decide the gut microbiota. If you eat more like fibers, you have different kind of firmicutes, that is kind of good bacteria. And if your food habits changes, then your bacteria changes over time, and that can lead to the inflammation in the gut. The interaction of micronutrients and microbiota is constant interaction and a very dynamic interaction with the immune system. And these immune system is continuously sensing these gut bacteria in the gut, and then actually shape whether to become pro-inflammatory or to become anti-inflammatory in this. So therefore, the IBD you can primarily categorize into two categories. One is the ulcerative colitis, is a disease of the large intestine, is a continuous disease. And the one is the Crohn's disease, which is a patchy disease, is a disease of the small intestine and causes ulcers in a kind of discontinuous fashion. And different mucosa, submucosas are affected in various these two conditions. So I will be primarily focusing on the ulcerative colitis. We have developed a model, animal model for ulcerative colitis. We call it oxazolone-induced colitis, and this is the model what I'm going to do. So first, I really wanted to give a sense what my lab is actually identified. We identify a distinct signature of TH9 cells. So what we have done, we isolated naive T cells from the mice, and then we sorted these cells based on the marker, and then we differentiated into TH9 cells, given the condition which is on the slide, TGF beta and IL-4. And these cells become IL-9 producing T cells. Then we actually subjected these cells into RNA sequencing analysis to identify the signature of these cells. So we found that the TH9 cells that produce interleukin 9 is having very distinct signature in terms of gene expression profile. And if you really go into profile, we found a lot of transcription factor or the factor which are upregulated or downregulated in these different conditions. And one of the factors which actually we focused on is the FOXO1 is found to be upregulated in the TH9 cells, and that is differentially expressed in these cell types. So what is the role of FOXO1 in these TH9 cells? We wanted to really understand, and then we did since this is a transcription factor, therefore we thought maybe it could be a transcription factor that actually drives the differentiation or drives the transactivation of IL-9 gene in these T helper cells. So therefore, what we have done, we actually screened the 2 kb proximal promoter of IL-9 gene, and we found the FOXO1 binding site in the IL-9 gene locus. And then we wanted to confirm whether this, what we found in the mouse, is also holds true in the human, and we found that the FOXO1 motif is actually conserved and preserved between the mouse and the human. And then we really went into identify whether whether the FOXO1 physically bind to IL-9, and then we did the chip PCR, and after chip PCR we found that we will able to enrich these 4 binding site. Out of 4 we able to enrich 3 binding site in the IL-9 locus in this. Therefore, we went into identify what if we delete FOXO1 in the CD4 cells using Cre-Lox system. So we use FOXO1 flox mice, and we cross it to the CD4 Cre, and then that means the FOXO1 will be deleted only in the CD4 cells, because FOXO1 complete knockout doesn't exist, because it's such essential gene. If you delete this FOXO1 in the whole body system, the mice die during the womb actually, so they don't actually come out from the mother. So therefore we use this conditional knockout approach. And what you can actually see, if you delete FOXO1, the IL-9 production is completely shut off from these settings. So therefore we concluded that this is a transcription factor which is important for IL-9 generation, and if you delete this FOXO1, it actually hampers or impair the function of TH9 cells and so on. And subsequently our lab is continuously working on FOXO1 and different, not only FOXO1 but the TH9 cells. We found EGFR, HIF1 pathway is important for TH9 cells, and very recently in the COVID-19 we found how IL-9 can actually aggravate SARS-CoV-2 infection and induces more severe allergic reaction in the lung. So in the lab, Rajdeep Dalal was one of the students at that time. Now he's a scientist in the lab, wanted to understand how, we can suppress IL-9 in inflammatory bowel disease, or in the condition by which we can suppress the inflammation in the gut. So one of the vitamins, we know retinoic acid is one of the retinoic acid, vitamin A. If you really take vitamin A, it actually suppresses inflammation. That has been shown. So we really wanted to understand what is the mechanism by which retinoic acid is able to suppress the intestinal inflammation. So these are the 2 questions really we asked in these settings. So first thing, we wanted to make sure what is the role of IL-9 in the ulcerative colitis. So you can clearly see you have a mouse model, which can be induce ulcerative colitis using this oxazolone, priming. And then in the one set of mice, what we did, we used anti-IL-9 treatment to actually neutralize interleukin-9. And what we could see actually, if you neutralize interleukin-9, the disease score actually much lesser. You compare the red versus green. And if you open up the mice and isolate the colon, you clearly see the lesion size of the colon is much higher in the oxazolone treatment as compared to IL-9 neutralization. So that actually gave us an indication that interleukin-9 could be a potential target for causing this inflammation in ulcerative colitis. And that is actually also kind of replicated here, that inflammation score by imaging is much lesser in the IL-9 treatment. The histology score, if you see this slide here, oxazolone, you see the very disturbed colon. There is no clear kind of villi, but if you treat anti-IL-9, there is a kind of reformation of these villi again. So that actually set it up the tone that we are really found that IL-9 is important. And second thing, when we started giving retinoic acid in one of the group of the mice, this retinoic acid is able to suppress the disease. You can clearly see the lesions are less if you treat the retinoic acid through the oral gavaging, and the histological score is also lesser in the retinoic acid treatment, like ulceration, inflammation, and edema. So now we found two things. One is IL-9 is important, and then retinoic acid is suppressing the inflammation. We asked the question whether retinoic acid is able to suppress IL-9 in vivo, and that's what we did. We collected the samples from the previous experiment, and we found if you treat mice with the retinoic acid in vivo, you see much lesser interleukin-9 and you have much lesser interleukin-9 in various organs, including mesenteric lymph node. So this actually set it up the tone that, two things, the association of IL-9 and retinoic acid and suppression of the ulcerative colitis. So we further wanted to understand what is the molecular pathway, or what is the molecule that is actually playing the role, because we wanted to go to the molecular level. So what we have done, we collected the samples, various samples, serum, stool, and tissue from the mice which either treated with the retinoic acid or not treated with the retinoic acid. And then we performed metabolomics profiling of these samples. And what we found, there is a differential regulation of different metabolites in the disease condition, either control, treatment, or the disease. When you say disease, it's ulcerative colitis. Treatment is the retinoic acid treatment of these animals. And one of the metabolites we found to be increased is the succinic acid, or we call it succinate, in the condition when you have more disease, and that is actually suppressed by retinoic acid treatment. So therefore, we thought maybe succinic acid or succinate is important. We also measured this in the stool samples, and we found in the retinoic acid treated stool sample, the succinic acid is actually much lesser. We also collected the lesion from the gut, and the lesion from the gut, place where there is no lesion and the lesion, and we found that succinic acid is much lesser in the non-inflamed as compared to inflamed tissue. We further went into the human settings, because most of the data what I presented in the last slide was the mouse data, but we wanted to make sure what we are doing actually also applicable to the human settings. So what we collected, Dr. Vineet Ahuja, I think he's sitting in the audience, or maybe he's going to give a talk today. He's a gastroenterologist. He runs the clinic in the Delhi AIIMS. So we collected the fecal material from him, from the healthy individuals as well as the ulcerative colitis patient. And what we found, we have more succinic acid in the ulcerative colitis patient. The most important thing I also want to emphasize here, that irrespective of different animal model like oxa colitis, DSS-induced colitis, or T-cell transfer colitis, the succinic acid is increased in all the different conditions, indicating that wherever you have the gut inflammation, somewhere succinic acid is involved or correlated with the outcome of the inflammation. So next thing, what we wanted to do, we wanted to really block succinic acid, because succinic acid is actually bind to its receptor. It's called succinic acid receptor. It's a GPCR receptor. So what we designed with our collaborator in that time, he was in the Punjab University, Dr. Deepak Salunke. He's a medicinal chemist. So he designed this molecule, or call it antagonist, for us. and this antagonist actually bind to succinic acid receptor. Therefore, the succinate is not able to bind to the receptor and or able to induce the function. And then we did an experiment, we induced colitis, and one of the group of the colitis is treated with this molecule called 7A, and we found there's kind of much lesser weight loss in this animal group. And you can also clearly see the lesion size is much lesser when we treat the mice with this succinic acid receptor antagonist. Similar kind of finding you can, when you collected the different sample, you say much lesser histopathology at different level when we treat the mice with this antagonist, and we found much lesser IL-9 when we treat this with the antagonist. So then we wanted to find a way how to increase succinic acid in the body. What is the way? And there are a couple of papers indicating that this fructooligoside saccharide, we call it FOS. If you eat FOS or if you give this FOS to animal, this somehow increases succinic acid in the animal models. And these are the two papers which I cited. And we did the same experiment. We induced colitis, and one group we gave this FOS actually, and we found FOS is able to increase more succinic acid in these animals. And if you induce colitis, you have much more colitis in the FOS group as compared to the control group. So that's actually further make a kind of correlation that this is really somewhere is somewhere is linked to the colitis severity. And when you have more FOS, you have more interleukin 9. So these things are kind of being connected. If you give more FOS, you have more succinic acid and you have more interleukin 9, and that may be causing the inflammation in the gut. Then we wanted to make sure in the gut you have different kind of T helper cells, and I wanted to make sure which of the T helper cells increase the highest level of succinic acid receptor. We did this differentiation of TH1, TH2, TH17, and we found that TH9 cells are expressing highest level of succinic acid receptor. And if you block TH9 cells with this antagonist, what I've shown before, is able to suppress the TH9 cell differentiation. Next, I can skip. So next, from where succinic acid actually comes in the body, it can be produced by two different ways. One is the oxidative phosphorylation or the respiration through the Krebs cycle. And this Krebs cycle succinate can be released. This is a cellular succinate. The second thing is the succinate can be produced by the gut microbiota. And gut microbiota produce large amount of succinate which is locally resides in the gut and can cause gut inflammation. So based on our actually pathway analysis, what we have done, and we found that the succinate what in our system in the animal models are not coming from the cellular respiration, it's coming from the gut microbiota. So next we wanted to make sure it is really coming from the gut microbiota. We generated these mice where we depleted all the gut microbiota with the 4 antibiotic treatment, as you can see on the slide. We took these mice, we depleted all the gut microbiota, we induced the colitis, and then we found that colitis by which retinoic acid was able to suppress otherwise in the wild type mice is not able to suppress. And similarly, you can see the colitis level is not able to suppress by retinoic acid if you remove gut microbiota in the mice. Then we did an experiment of this called FMT. So we collected the fecal material from the two group of mice. One is the oxazolone colitis mice, one of the oxazolone colitis treated with retinoic acid. Then we actually gavage these gut microbiota in the healthy mice, and then we induced the disease. We wanted to make sure whether the gut microbiota coming from the retinoic acid treatment is able to transfer the protection in the colitis, and that's what we actually found to see. In the gut microbiota coming from the FMT coming from the retinoic acid recipient is able to suppress colitis as compared to the oxazolone. So that actually gave us indication that this is really happening through the gut microbiota, and you can clearly see whenever you have done the FMT, the retinoic acid FMT is able to suppress IL-9 and IL-9 receptor as compared to the FMT from the oxazolone-induced mice. So next we wanted to really go directly into the, we don't want to beat around the bush. So we asked the question, let's do the sequencing, 16S RRN sequencing in the system, what we are doing, and whether we found some gut microbiota which is kind of upregulated and which is kind of inducing protection. As clearly see here, you have two different treatment, oxazolone colitis with retinoic acid. We did the sequencing and we found some of the family of the bacteria is found to be upregulated when you have the oxocolitis, or found to be downregulated with the retinoic acid. One of the things, if you really notice here, Bacteroides vulgatus is one of the bacteria found to be upregulated in the oxazolone colitis. And if you really go and do the research on this bacteria is the highest succinate producer in the gut. So it produces large amount of succinate in the gut. And what it actually bacteria does, if you see the treatment with the retinoic acid, it actually goes down, because this vulgatus is also having a retinol sensor into it. So the moment it actually get exposure from the retinoic acid, the retinol sensor in the bacteria allow this bacteria to kill itself. So therefore, the retinoic acid is able to suppress this bacteria as compared to this oxazolone. And then what we actually did, we asked the question, can we put the vulgatus and see whether vulgatus can severely induce this colitis? And then we colonize the single bacteria, isolated vulgatus, and then vulgatus is able to induce more colitis as compared to oxazolone. But when you treat with the anti-IL-9 or 7A, because these are the effector molecule, the vulgatus induced colitis is actually reduced completely in these groups. And same thing actually you can clearly see, with the vulgatus you have more IL-9, right, as compared to oxazolone, and if you remove these effector molecule, is actually suppressed. How many minutes I have actually?
24 seconds, but sir, you can.
Okay, I just maybe have 3-4 slides. So next, we really wanted to understand. We went into the literature and we found that there are kind of bacteria in the gut. Those are we call succinate producer. They produce succinate. At the same time, you have succinate consumer. So basically, in the homeostasis condition, you have succinate which is being produced in the gut, and it is being also consumed by the bacteria and convert into propionic acid. So when you have homeostasis condition, you are very healthy. You see more propionic acid in your fecal material. That means you are healthy gut, right? The moment you have more succinate is coming in your fecal material, that means your gut is inflamed. And this is somewhere your balance between gut, the succinate producer versus succinate consumer is disbalanced, and that may be causing the inflammation. So this is exactly we did. We found with the retinoic acid, we found more succinate consumers are being upregulated in the gut, as you can clearly see by this. You have different Phascobacterium, Faecium, diasteral species. So we identify many bacteria which is consume succinate and break down succinate, and those are upregulated by the retinoic acid treatment. So then what we did after searching all this literature and our own sequencing data, what we also did, we isolated these single bacteria. You can see these are the 4 different bacteria which isolated. These are the succinate consuming bacteria. And after isolating these bacteria, we did again the same experiment. We put these bacteria colonize in the mice, and then individually we colonize these bacteria in the mice and induce colitis. And we found that if you colonize these bacteria, they are able to suppress colitis in these settings. Now we also made this, again, you can clearly see the succinate consuming bacteria, you have more colitis lesions, and if you put this succinate consuming bacteria, there's no lesions, the colitis score much lesser, IL-9 is much lesser. So that is indicating that if you somehow use this bacteria, you will be able to suppress colitis. And what we did, we also make the consortium of these bacteria. We made the consortium of these bacteria. We combined these 4 or 5 bacteria to make a consortium, and we made this patent as a Indian patent. And now we are trying to see whether we can generate the GMP tablet of these, spores of these bacteria, and when can we go to the clinic of IBD. And one more thing, actually, I want to also emphasize, which I did not really show the data here, that the succinic acid is also found to be upregulated in the type 2 diabetes. And we have done the experiment in the type 2 diabetes in the animal models. We have given the weekly basis the oral gavage of these consortia, what we identified and made, and we found the glucose level in the animals as compared to control is going down. So this could be another indication by which the consortia tablet, what we are proposing, could be beneficial not only for inflammatory bowel disease, but for other metabolic diseases, including type 2 diabetes. With this, I would like to conclude, and these are the people who are contributed. Rajdeep Dalal, who is in the audience, and you can have questions with him. He has a lot of great work. The funding agencies, this is my lab. Dr. Vineet Ahuja, perhaps is going to give a talk this afternoon. Saurabh Kedia and all funding agency. Thank you very much. I will be happy to answer the questions.
Thank you very much. Hello. Thank you very much, Dr. Awasthi. Now, we are really enchanted to hear your talk. Now, this problem, this gut microbiome, butyrate producing taxane, its importance is there now in student syllabus. Now, students must be thinking now, they are sitting at back. Now, Professor Awasthi is adding one more, the succinate producing one. But it was going to come. So, it was very clear. I think everybody has understood and everybody have appreciated. And I was seeing the students' response. So I think they are thinking that from this butyrate producing, now they have to do student experiments on succinate producing or estimating not only propionate and SCFAs, now it's the era of succinate. So it was that much clear. So I open this to this esteemed audience for interaction, please.
Hi, excellent talk, and really it was very nice. I'm Dr. Manu. So my just question is that in the experiment which you have used B. vulgatus, right? So in that you did not use retinoic acid treatment, right? So the reason for that, or is it that you didn't do it or did not give you the proper
No, I actually did not show the data. It's a part of the manuscript. As I mentioned to you that the vulgatus is having a retinol sensor into it. Yeah. So when you colonize the vulgatus, they produce more succinate. But when you give the treatment of retinoic acid, the retinoic acid being sensed by the bacteria and its growth goes down. So then it actually suppresses the abundance of the vulgatus, and as a result, your inflammation score gets better.
But you use the retinoic, the drug which you have synthesized, right? That
no, so we did
not add retinoic acid.
We did multiple things. I did not show the.
You did not show the data. Okay,
so,
that's right.
Yeah, the retinoic acid data I did not show, but I show the data of the anti-IL-9 and the 7A here with the vulgatus.
Sir,
Please.
please. Here. This is Prakash Alami from CFTRI. We use Bacillus and Bifidobacterial probiotics for treating ulcerative colitis. Since Bacillus they colonizes throughout the gastrointestinal tract and Bifidobacteria being in the colonic regions. So how effective you consider over FMT or the bacterioid treatment?
So one of the major challenge in the field of microbiota, when you are experimentally, you can implant single bacteria because these are the kind of closed conditions, very nice. But some of the things which is important for bacteria to help and colonize. Therefore, in our consortia, we added some of the bacteria which actually create a milieu to help them colonize it. So that will be maybe better through the oral space because we created an environment by which the other bacteria, which is succinate consumer, is going to be colonized. That is a part of the consortium.
You suggest bacterioid to be used being a gram negative.
So bacteroids is coming in many things. Even in the anti-cancer, it is coming bacteroid. We have a paper in the Science Advance. That is, but there are multiple things we have to first check with other conditions. You know, if it is working because ultimately what is happening. It is conditioning the immune system, and immune system is a double-edged sword. It can suppress something, then it can increase something, right? So we have to do more experimentation to identify really what is the effect.
See, in our attempt to reduce the amount of sweeteners in products, instead of added sugar, now we are replacing with phos as a sweetener. Now, looking at your data, it could be a matter of concern for people to consume products having phos. Maybe we'll have to have a warning label on these products because it could aggravate the situation in some of the consumers.
Yeah, definitely. So when we were doing these experiment, and obviously the sweeteners or the kind of sweet thing is increasing a lot of succinate, that is known, and a lot of papers. But when we were doing this phos, there was a couple of papers what we mentioned, and we were kind of astonished to see that, you know, it is able to increase gut inflammation. So this could be maybe the phos product. I really cannot really claim because at this point, because these are the manuscripts should come out, but that label warning should be there on the phos product. It's not really very good for the gut. It may be good for other things, but maybe not good for the gut. That's what we feel.
Dr. Awasthi, thank you for a very nice presentation. I am Dr. Sarat Gopalan. I'm a pediatric gastroenterologist from New Delhi. Purely out of curiosity, how imperative is our need for a stool repository, a stool bank, like we have our human milk banks and blood bank, in the light of FMT? Is there any progress in this regard for India coming out with a stool bank? Or
I think Dr. Vineet will able to answer this question, but I know one of my friend who is based in the Texas, MD Anderson. He started a company in India. The whole idea to collect the so-called healthy stool samples, because ultimately, if we have to go for the FMT, right? So therefore, if you create a bank as a healthy stool sample, that is how his company is doing in India. And Dr. Vineet is also somewhere. Dr. Vineet is here. I think he can maybe answer this question. Yeah. Because here, Dr. Vineet has done a lot of FMT. I mean, his paper came out very nice, so he can answer about the bank.
Thank you, Amit. That was a very stimulating talk. So for stool banks, the thing is that now I think we have crossed that stage of stool banks. Now it's a question of making capsules. Now the thing is you make capsulized FMT in the capsulized form, again, where you have the microbial consortiums, or you may also have the metabolite consortium, like Amit showed you succinate. So you can make metabolites, and you don't, the chairperson talked about short chain fatty acids. So you can make a capsule of short chain fatty acid as well as succinate, if both are going to be beneficial, by only one constituent. Similarly, for stool banks, the question is that, okay, in India, there has always been an issue also about polio carriage and all those kind of things. But more importantly, that you screen the donors, and subsequently, if you take them into a capsulized form, it becomes a homogeneous form. It becomes a something whereby it goes into the drug category, so that it is reproducible. You know exactly what the constituents, and at the same time, there are no adverse events related with it.
Mic to
Dr. Ganguly,
please.
Hello. I've got a question and I've got a mic already. It's Patricia Conway from Singapore. I was a bit intrigued by the FOSS story, considering that FOSS-GOS mixture has been added to infant formula for years now. So it's a short chain GOS and a long chain FOSS. Is this of concern with rising inflammatory conditions in children that are fed infant formula?
So this is regarding the FOSS. Can you repeat the
question? Okay. Yes, it's with regards to FOSS and the succinate levels.
Yeah.
And there's a lot of FOSS added to several infant formulas.
Yeah. So this is again same questions what you are kind of asking, but it's a different thing. It's the product with the children. Obviously, I think the FOSS in our study and some other study is able to increase the succinic acid level. And what we are showing that increased succinic acid level through the FOSS is able to make your T cells become more pathogenic. So obviously, I mean, these products whatsoever using the FOSS, more data is required, right? And therefore, it has to be like kind of tell the regulators that this may not be healthy things. Because ultimately, if you really change the gut microbiota in the children, it's going to affect long-term effect actually.
Amit, one of the short points which I want to make is this, that the whole microbiome situation is not that simple. Like we heard one of the talks today about LCS and a particular signature in LCS which makes it LCS. When we have done the gut microbiome transplantation from the very normal donors, there are only few who becomes super donors, that they survive for a long term and change the metabolism effectively. So I think we should, like Akamencia was one of the things which was shown today. So we need to identify such bugs, find signatures, but ultimately when you are thinking of tablet, there will be one more talk from Osaka University today. That if it is a mixture of a symbiotic and the bug, it might work better. So just bug may not be able to do it. The bug, like if you want to grow the bug, you have to provide carbon source for fermentation, other things. So you may consider to find the signatures which will actually will move metabolism. And one of the things you had yourself isolated and seen that how it works. So my suggestion is that there's a beautiful hypothesis, and we need to go much more in depth with some of the bugs which you have isolated.
Thank you, sir. I totally agree that single bacteria may not be a solution when we are going to an outbred population like human. In the controlled environment in the animal, can be done. But that's why we are more focusing on the consortia of the bacteria, not a single bacteria or two bacteria. I think if you increase the consortia, you have more diversity, and that some of them may be not able to colonize, but other may colonize and help other them to colonize. That's the one way to go. The second way, I think people should also are talking about the kind of superfood nowadays. So if you take these bacteria and then you go on to the diet, which is anti-inflammatory diet, so that these bacteria, that diet will help you to colonize these bacteria faster. So I think ultimately it's going to be combination of multiple things. It's not going to be one thing. Yeah.
Another problem with the combination is, the biggest problem with the combination is that some of the bacteria, they reciprocally regulate each other.
Yeah.
Dr. Awasthi, just a thought on the FOS story, because FOS, as we all know, is a very well recognized prebiotic. Maybe the concentration of the FOS could also be responsible for the production of succinate.
So that is, I think, very, very good point what you are making. And the concentration what we have used is based on the concentration what is kind of published. So we did not really go into the kind of different doses and see the level of succinate and whether lower dose of FOS is not able to increase the inflammation, all those kind of. I think that's a very good point. I think we will be able to do an experiment on this. Yeah.
Any more questions? Hello. Any more questions? I think this question session will not stop. So it's please.
Hello, can you hear me?
Please introduce.
So, we have been talking about, it's a very good presentation, but what I want to say is that there are many, other volatile fatty acids, and it is the balance between the two, whether it is succinate, propionate, or other volatile fatty acid. So we have to have that kind of conditions where we can, use multiple organisms, which you are yourself saying, and then prove the point. Because now with this data, if you go and stop giving FOS, I think that's a matter of concern. So we have to have a real situation, because it may be different in a mice or in a model which has been artificially created. But if you go to patients, the story is very different, and it is multifactorial, and there's so many organisms there. So I think it requires, more thought-provoking experiments and more data and more in vivo data to take the process further.
I totally agree. I think regarding the FOS story, we rather go directly to the human samples, like fecal material, the people who are on the kind of having this kind of, sweetener or the FOS, as compared to the population which is not. So then we can measure some of these, succinic acid or other short chain fatty acid in those, and see people who are taking real time dose of FOS, what is the level of the short chain fatty acid in their in the gut.
I think if there is more question, it can be dealt at other times, at the meeting times. So now, thank you very much, Dr. Awasthi.
Thank you.
It's a very nice talk. Big hand for him, please.